Thank you, Alex, for the introduction, and welcome back to the session. So a very good morning to everyone, and thanks to the organizers for putting together such a nice program, and also giving me the opportunity to share some of our group's recent work relating to wearable on-skin chemo-sensors. And before the break, Aaron just excitedly announced that the conference is going to move to Singapore next year, and so a free welcome to everyone. Incidentally, I'm also from Singapore. I'm in the Institute of Materials Research and Engineering there, of A-Star in Singapore. So it's basically a collection of national labs and research institutes, and it's located very near to the National University where the next flex will be held at. So, welcome in advance. I'll just briefly introduce my research group. I lead my research lab called the Profess group there. My name is Le, by the way. Profess stands for your Printed Organic Flexible Electronics and Sensors group, and we mainly work on two different topics. So on the right, we are looking at a lot of organic semiconductors, organic luminescent materials, and optoelectronic devices. And recently, we are looking a lot on photon-up conversion, in particular, looking at the process of triplet annihilation in organic semiconductors used in photon-up conversion. And so we've got a couple of maybe I'll just stand more in the middle and point to this. We've got a couple of recent publications on this topic if it's of your interest. Along with photon-up conversion, we are also looking at other electronic devices, including OLEDs and electrochemically excited luminescent devices. For today's talk, we are going to look at the other portion, which is on wearable technologies and flexible biosensors. So before we look at what we do in our group, I'll just bring us very briefly through a backdrop of the field, which really, blossomed in the past decade or so. Some of the, milestone works are shown here. Of course, they're not exhaustive. There are many other very notable, significant works. We're showing a few here, whether they are, based on electrochemical methods of detection, or at the bottom based on colorimetric methods of detection. But they're all looking at how to detect chemical concentrations from sweat, from the bodily fluid of sweat, which is very readily available when you make it into a wearable form factor. So what we want to bring to the table, what we want to emphasize is that it comes at two levels. So the first level is more on translation, right? How can we make something that's more scalable, something more fully printable, so that when we develop an integrated wearable device, it can be more suitable or compatible with downstream translation. And on the second level, it's slightly more fundamental. What if there is no sweat, right? What if we don't have that readily bodily fluid on dry skins like now? In Finland, we're not sweating so much as in Singapore. So what if we wanted to work on everyone else? So these are the questions that we want to answer, and of course, along with that, other things like it has to be multiplexed, truly wearable, truly user-friendly, and very importantly, it has to be correlated to some kind of health parameters to indicate our medical conditions, right? So given that backdrop, the outline of my talk would be, we'll start by looking at the sweat sensors from our group first. So we call them the WISH devices. I like acronyms if you realize my group is called Profess, and our sweat devices are called WISH devices. So they're wearable in-situ sweat sensors. And so when we look at, most of the developed countries around us, we usually estimate that about 30% of the population are regularly active, regularly sporty and sweaty. So based on this, we can target this population for more sweaty activities based on the bodily fluid of sweat. Right, and of course, we are looking at building WISH devices which are non-invasive and in-situ detection, and they are also made in a printed and flexible manner. So we'll start by looking at a colorimetric WISH device, and that's more for non-continuous and time-pointed single measurement kind of use cases. But that's not good enough, of course. We then move on to electrochemical WISH devices, which enable us to do a more continuous kind of measurement. So at least you can track the changes automatically, passively, over a few hours of your being sport and having sweat, right? And we also know that that's still not good enough. What if we want to access the rest of the population who are not regularly, sweaty, basically, or you have a medical conditions to look at, that doesn't allow you to sweat so easily, right? So then here we are looking at accessing the rest of the population, and that means we need to develop some chemical sensing that can happen without fluidic environment, without sweat. So we will showcase how we can enable that technology, as well as some correlation with blood biomarkers as well. And lastly, we also illustrate how to make hybrid zymes, which are an enzyme complex and material-based method that can enable us to have a very stable, sensitive, and selective devices. Because now we are talking about long-term wearing, long-term usage. We're not only looking at sporty activities only for a couple of hours of sweating. We want the user to wear it for a longer time and a very stable, capable device if needed. So that's the journey that will bring us through today. And of course, we'll start from the wish devices, the sweat sensors first. Right, so first up is our colorimetric wish device. This is a wearable, paper-based, fully printed, sweat-based colorimetric sensor. So we've developed a ceramic-based ink that we can then print it out by an automatic ink dispenser. Print out the zones, the boundaries of the zones and the channels that we want to design. And after that, we functionalize the detection zones in the channels. So as we can see, for example, here we've got 4 different detection zones, each for a different marker, as well as a moisture-sensitive channel, the coloration of which will tell us the sweat rate of the person. Right, so this whole device, as you can see, it's not electronic. It's very small, thin, flexible. You can easily stick it on your skin. So, and this is mainly done by this novel ink that's based on a barium titanate paste together with graphite powders. And also we mix in organic polymers to tune its viscosity during printing. And so compared to conventional methods of making paper fluidic colorimetric devices, compared to conventional ways of making similar printable devices, those are usually done by wax printing. So compared to the wax materials used for those printing, our ink basically show quite a bit of enhancement on a few fronts, including the pattern resolution, the way sweat seep out or don't seep out from the boundaries of our printed patterns, and also its stability towards heat, stress, and harsh chemical environments. So we see an improvement on all these fronts. And here we are showing visually a short video. Basically, this is the automatic ink dispenser that dispenses our ceramic-based ink and print out the template basically of our structure. So those are the 4 detection zones, and now it's printing the sweat channel that's drawn now. So I won't show you the whole video. That's the idea basically. So another challenge is the color homogeneity of our sensor within the confinement of the zones. Because our paper is a cellulose matrix, right? It's very porous. So the reagents and the dyes will be quite mobile within that zone, and it would tend to run to the edges of the zone, and that leads to inhomogeneous coloration. So in order to counter that, we then functionalize the zones with nanoparticles, with aiming functionalized nanoparticles, and that helps to fill in the pores of the paper and confine our dyes and the reagents and immobilize it evenly across that area so that the coloration is more evenly distributed, and that helps with the sensitivity as well. And so lastly, we put it on body. Some real people running, sweating, and we look at the accuracy between what we can measure on body versus a commercial assay kits. And we can see that the bar charts are very closely together. So that's a very good agreement in terms of accuracy validation there. And so that brings us to the first part, which is the colorimetric wish devices, which, as we can see, is a time-pointed thermal measurement, a non-continuous way of looking at sweat chemicals, right? So then let's move on to the next improvement, which looks at a continuous way to measure to look at chemicals in sweat, and that's by the electrochemical method of detection for our WISH devices. Right. So this is still unpublished, but basically this slide shows our work stream all the way from formulating our conductive ink that can be then printed out by screen printing. We chose screen printing as a more compatible way to scale up and also downstream translation as well. So we use our ink is based on graphite, by the way, it's carbon based. So on the bare electrodes that we screen printed out, we then functionalize each working electrode with the specific enzymes and nanomaterials so that they become a targeted sensor towards a specific analyte. And lastly, well, you can see here that we have 4 working electrodes multiplexed together onto one device, and that one device is then integrated together with the PCB board, power, the battery, the Bluetooth. There's also an app for the user to non-invasively and in situ continuously monitor, look at the readouts, right? So that's the eventual integration here. And so again, I have a very short video. Okay, this is showing the screen printing process of our carbon-based ink. And as you can see, on one piece of a flexible sheet, you can have many pieces printed out. We can then cut out the individual sheets. And on each device, we have 4 different working electrodes multiplexed already. Just now we saw the benchtop functionalization of each of the working electrodes. And here we see that the sensor is plugged into the PCB, connected to the wire, the power, and is transmitting in real time wirelessly to the app. That was some benchtop testing of dropping of spiked solutions. And lastly, we do it more real life. We put it on our colleague who's running and sweating, and you can see accordingly the changes happening in real time as well. So a few of our team members have been repeatedly going on about helping us with the running trials, and our team in general is getting healthier and slimmer as the project goes by. So that's one good thing coming out of it. So yeah, that's visually how it looks like. This is a deconstructed view of that little plastic strip of sensor that we saw just now, right? So basically it's a layer by layer, bottom up kind of architecture. What I want to just highlight is the top layer. We call it the paper kirigami sweat channel. So now if you imagine we're talking about continuous in-situ sweat cleansing, that means we need a mechanism to continuously bring fresh sweat from our body, from our skin, to the sensor and actively bring it away as well. So there's a fresh flux, fresh flow of new sweat, so that we're not reading old accumulated pool of sweat, right? So we developed this way of making very cheap, easy sweat channels by taking a piece of cellulose paper, cut and fold, hence kirigami, into something like this that can wrap around our sensor pads. And that forms a paper channel where a sweat is being sucked in from the bottom, travels through the channel, and goes out by passive evaporation from the top. And that flow rate through the paper channel is fast enough to keep up with the actively exercising sweating person. And so all our on-body trials have been done using these kind of paper sweat channel, kirigami sweat channels in tandem with our sweat sensors. And so I mentioned we did a lot of on-body trials. We're also looking at some of the dietary effects and exhaustion-related effects when someone runs for a really long time. And we're preparing the publication now, and hopefully we are able to share more details from both the device and the data soon. So that's our sweat-based devices. And as I mentioned, that's not good enough, right? Like we need to accept the rest of the population who are not regularly sweaty and sporty, or they don't live in hot countries, or they're medically ill or are unable to sweat so much. And with that, we then move on to the second part of our talk today to achieve that, right? So here here we're looking at basically our goal is to develop an anytime, anywhere, anytime kind of wearable sensor that can enable a non-invasive, continuous, in-situ chemical monitoring from our body. And there are 2 main challenges that we want to tackle here, and I'll go through both of these in the second part of the talk. So the first challenge is that chemical sensing is very difficult to happen without fluid environment. That's why we always need a bodily fluid for it to happen, right? So, but if you imagine a solid state analyte, a dry analyte versus against a solid state electrode, there are no ionic or electronic conductive pathways, and therefore that's why the chemical sensing is really difficult. And here we have therefore devised a solution, an interface to allow such an interaction to happen. And the second challenge has got to do with the intrinsic enzyme instability, the enzyme denaturation, that makes enzyme-based biosensors kind of not so long-lasting, and we will then share our innovation on how we counter that problems using a materials-based approach. So going to the first one first, as I mentioned, fluidics, non-fluidics environments for chemical sensing very difficult, right? So what we do is that we have developed an interface that consists of a bilayer of hydrogels that forms this solvation diffusion function kind of layer. So bilayers, so two layers. The first layer is the ionic conductive hydrogel, and that's in contact with our skin directly. So the solid state analytes from our skin surface will dissolve into that gel and diffuse across the gel towards the second layer of hydrogel, and that's our electronic conductive hydrogel, the ECH. So our analytes diffuse toward that and reach that interface. Our electronic conductive hydrogel is basically our electrode that's already been functionalized with enzymes and, well, where the actual sensing happens, actual electrochemical reactions happens. So the analytes diffuse from the skin across the ionic conductive hydrogel towards the interface to be sensed, to be detected. So that's the idea behind the whole work, really. And as you can see, our overall form factor of the device is very thin, plaster-like, flexible. It's stretchable as well in this variation. And it can be easily stuck to the skin, and it's also fully printed by printed fabrication route. And let's see. Okay, yes. So for this, for these purposes, we are looking at solid-state epidermal markers, and we are developing a sensor for that, right? And that's why we call it SEBS, the solid-state epidermal biomarker sensors. And for the SEBS, we are demonstrating two different biomarkers on this platform. One is the water-soluble lactate, and the other is the water-insoluble cholesterol. And so for each of these cases, the ICH, the ionic conductive hydrogel layer, has to be carefully tuned and optimized so as to promote the maximum, the best way to dissolve our target analyte and let them diffuse through that gel. Right, so here we show some of the basic electrochemical characterization, and I'll just highlight that this figure here shows the selectivity is highly selective and specific towards the targeted analyte, and that's very much helped by our ionic conductive hydrogel, which is very porous in nature, and it helps to block out a lot of large macromolecules, proteins, to stop them from interfering with the measurements. And lastly, here, these are some of the accuracy validation. So we test our devices against same samples measured by commercial assay kits, and the linearity confers accurate validation. So in terms of a bit more details here, in terms of mechanical, it's very mechanically robust as well as stretchable. So it conforms to skin quite well, very comfortable to wear, and there is no skin irritation of prolonged wearing. It's also stable against motion. And over here, the bottom figures, they are simulations to illustrate that the entire sensing the response time is very fast. The entire sensing process can be completed within a few minutes, and that's suitable for a continuous in-situ real-time applications. Now we look at something a bit more visually stimulating. This is how it looks like when it's worn on skin. The user can wear it and goes about activities of daily living, ADLs. And here we look at a bit of dietary effects. He drinks a high sugared drink, And after some time, this is showing lactate reading. The lactate actually goes up quite a bit. There's a time lag, of course, for the skin to show this. And that video brings us to the exciting part, which is also the subject protocols. And we put it on quite a few people to see how it works. So basically, we are looking at someone who's under fasting versus after having a meal for a couple of hours, and we can see there is indeed a change between the cholesterol and the lactate levels. And more excitingly, we then correlated the on-skin readings with blood serum biomarker concentrations as well. So here, if you look at this figure, we are testing across quite a few hours through the day. In the middle, the user takes a meal at a time-pointed event, at the same time points that we take a on-skin reading, we then also do a fingerprint test to look at the serum concentrations, right? And we can see that the trend follows very well, and the linearity or the correlation between the solid-state epidermal markers versus the serum markers are really high. And that, gave us a very good confidence that, solid-state epidermal markers could be a source of indication for our health parameters, because we know blood is the gold standard already, right? So we're still carrying on this work to push it further, but that was recently published as well. So that's the enabling of the dry skin sensing, basically, right? And next, we're looking at the stability. Now that we can have the user to wear it on dry skin for not for sweaty purposes anymore. So we want the device to last as long as possible. So for biosensors which are based on enzymes, now enzymes are proteins which are held together by chemical bonds to create that specific 3D shape and configuration that confers its catalytic functions, right? So under stress or heat, under electrochemical testing, for example, the bonds would break and the protein would unfold or denature, and that leads to a loss of the specific shape that's held in place, and therefore its catalytic functions as well. And that's basically the problem intrinsically with enzyme-based application, that the stability suffers. But wild-type enzymes, they have very good specificity and sensitivity for devices. And there's always seems to be a trade-off when people try to improve the stability. There is a suffering of, there's usually a suffering of sensitivity and efficacy in general. So we try to then overcome this compromise, overcome this trade-off, right, by maintaining its sensitivity while prolonging its stability, right? So what we did is that from a materials point of view, we co-encapsulated the enzymes with a carbon dot, with a type of carbon dot in a mesoporous morph network, metal organic framework network. And that leads us to have a very highly stable nanocomposite, what we call a hybrid zyme complex, a hybridized enzyme, basically. And with the hybrid zyme, we then made electrochemical sensors that we fashioned into a fingertip touch-based glucosal lactate sensor. And that basically gave us, the holy trinity, the stable, selective, and sensitive 3S cis all-in-one kind of device that we're quite happy with. So why does it help with such performance, right? So we did quite a bit of characterization, and we suggest that it could come from 4 main factors for the improvement, the simultaneous improvement of sensitivity and stability. Right, now the first one is that by co-doping the carbon dots with our enzymes, we somehow end up with an optimal nanocomposite sizes that are ideal for such sensing. And the second thing is that the carbon, by having, co-encapsulating or co-doping the carbon dots into the morph network, the pores of the network are larger than before, and that helps with analyte diffusion. All right, coming to the end soon. Make the pores larger so that the analytes are easier to diffuse and access the enzymes. And the third thing is that the carbon dots also have a peroxidase mimicking property. So it helps with the downstream catalysis of the H2O to hydrogen peroxide in that enzymatic cascade reactions. And lastly, there's the tiny little carbon dots might also bind to the enzymes and stabilize the structure, so it prevents it from unfolding and denaturing that easily. Right, so that's, well, let's just take a brief look at the performance, right? So for most of the paper, we did on the model system glucose oxidase, but we also did on a secondary work, we did work on secondary enzyme lactate oxidase. And basically, repeatedly, we see a 40% sensitivity improvement, and we can look at this hybrid design approach as a platform strategy towards improving enzyme characteristics. Not just sensitivity, but very importantly, the stability for long-term use, right? So whether it's under high thermal stress or harsh chemical environments, even at elevated temperatures of nearly of around body temperature for up to a month or more than a month, really, it's still showing an improved, consistently improved sensitivity performance. I mean. Lastly, so we made it into a touch-based fingertip chemical sensor, and again, so we use the same device repeatedly over a month, and we see very consistent readings from that same device the whole month over the whole of 30 days. And lastly, I'll just very quickly show the video of how it looks like. That's our sensor. Now we're putting a hydrogel on it. So this is also a hydrogel-based interfacing with our skin. But at this stage of our work, we haven't developed the fully solid-state dry skin version yet. So for this hydrogel, for this hydrogel version, we still rely on a tiny little bit of sweat from the fingertip for sensing to occur. So it's an in-between state. So anyway, we put the finger onto the gel for a few minutes for the sweat to be concentrated into the gel, sequestered into the gel, and that's where the electrochemical reaction or sensing happens. And so with that, I will just conclude and summarize my talk. So we've seen 4 different areas of work, and we've brought us through this journey from a more primitive sweat sensor all the way to something that's more applicable across a wide range of applications and population. And with that, I think, yes, that's the last slide. I thank you for your attention, and also my team. We've talked about biosensors here today, our team, and also our collaborators, some of whom are in the conference or involved in the conference. Aaron and Zhang Jian is also part of the committee, I think, for this conference. And also I thank my funding agency as well. Thank you.